part 2. So in part 1, we went over a diagram of a generalized bacterium, and now we're just going to take a look at each of those structures in a little bit more detail. So we'll first start out with the living boundary of the cell, the plasma membrane, also known as the cytoplasmic membrane or cell membrane. The most important job of the cell membrane is to control the movement of substances into and out of the cell. So the structure of the cell membrane, we use a fluid mosaic model where we have an ocean of phospholipid bilayer fluid. The phospholipids can move side to side. And then I think of the mosaic tiles as being the membrane proteins. And we'll see one of the function of membrane proteins is to help large hydrophilic molecules to cross from one side of the membrane to the other. Now, very important for us as microbiologists is that if anything damages the cell membrane, that will kill the cell. And we're going to use that to our advantage. When we have bacteria, pathogenic bacteria, growing within our patients, we want to kill them. We can use antibiotics that specifically target and cause damage to the cell membrane. And these are just 2 examples, folks. I never asked these on the first lecture exam, but just so you know, polymyxin B and daptomycin can either damage or interrupt proper function of the cell membrane. Again, apologies for the poor quality of these slides, but I wanted to make sure that you felt comfortable with the diagrams that we'll pass out in lecture. We'll give you cell diagrams. And the reason for this is often these diagrams or diagrams similar to these will appear in the lecture exam. So I wanted to make sure that you felt comfortable interpreting them. So these are 2 cartoons of cell membranes. So this one is showing us this little guy right here is a phospholipid, and you'll recall we use the circle for the hydrophilic heads and the 2 lines for the hydrophobic tail. So remember that phospholipids are amphiphilic or amphipathic, and when we mix them in with water, they can spontaneously form these really cool phospholipid bilayers. So do remember that the hydrophilic heads will orient so they're interacting with water on this surface and that surface. And then do remember, folks, be able to label this is the hydrophobic region where those hydrocarbon tails are located. And this helps us understand why a phospholipid bilayer is selectively or semi-permeable. If we have a large hydrophilic substance here, there's nothing to draw the hydrophilic substance across that hydrophobic region right there, because the hydrophilic molecule will want to associate with water, or it'll want to hang out with the hydrophilic heads. There's nothing to draw it across this hydrophobic region. And the same would be true if we had a large hydrophilic molecule here on the other side of our phospholipid bilayer. There's nothing to draw it across. So we would say phospholipid bilayers are going to prevent passage of large hydrophilic molecules. And then that will be the job of many of our membrane proteins, such as this one here, will be to help large hydrophilic molecules cross from outside the cell to in, or from inside to out. Now, in this little membrane protein here, this little globular membrane protein, you can just barely make out there's some charges here and there. And this is kind of cool because it helps us to understand that our globular proteins are going to, they'll fold so that they're polar or ionized amino acid residues with polar or ionized R groups will be on the outside here and there, again interacting with water, and the amino acid residues with nonpolar hydrophobic R groups are going to get pushed into the middle of the protein right in here. And then this is just a nicer picture of a cell membrane. So again, folks, this is a phospholipid bilayer. It's fluid. These phospholipids can move to and fro, to and fro. That membrane fluidity is really important. The membrane has to remain fluid without melting. And then what are these in here? Come here. Okay, so here's a protein, right? And what would be one function of this protein, say? Right? This could be a transport protein assisting large hydrophilic molecules to cross. And we know that this protein would then be made up of amino acids, right? Our phospholipids are going to be made up of fatty acids, a glycerol backbone, phosphate, and an additional organic group. Okay. So just a little bit more detail. So again, our phospholipids, hydrophilic heads, hydrophobic tails, forming the bilayer here. Here's our ocean, our fluid ocean of phospholipids and our membrane proteins. And we can see here, right, this would be a water-filled channel through which hydrophilic substances could pass. And again, that phospholipid bilayer with proteins will form the cell membrane, the cytoplasmic membrane, plasma membrane. And do notice it's pretty delicate, right? Okay. So those membranes, if the phospholipid bilayer has a consistency of olive oil, you can imagine they're really quite delicate, right? And so we want to recognize that those cell membranes, cytoplasmic membranes of most bacteria, they're not strong enough to prevent a harmful process called osmoticlysis. So we next want to explore, what do we mean by osmoticlysis? So osmotic sounds like osmosis, the movement, the diffusion of water from an area of high concentration of water to an area of low concentration of water across a semi-permeable membrane, and lysis is to break open. So let's check this process out. This is kind of the classic cartoon that we use to describe osmosis. Again, movement of water from an area with higher water concentration to an area with lower water concentration across a semi-permeable membrane. So in these YouTube experiments, this would be our semi-permeable membrane. It permits water to cross back and forth, but it wouldn't permit larger solute molecules to move back and forth. So what we want to do is compare the solute concentrations on either side of our membrane here. So the red dots are solutes, and so you can see we have more solutes, higher concentration of solutes on this side than that side. Now the solutes actually take up room. So if we have more solutes, that means we have less water on this side, and vice versa over on the other side. Since there's fewer solutes, that means there's more water. So we'd have a higher concentration of water on the right side than the left side. So we're going to see diffusion of water then from the right side across the membrane to the left side, right? And that's osmosis, right? And so here we see that we have water flowing over from right to left. Now let's apply that to our cells, okay? So the problem is most cells are living in what we call a hypoosmotic environment. That means the concentration of solutes outside the cell is lower than inside the cell. And this is often because the cell is actively transporting nutrient molecules into the cell. So again, folks, a hypoosmotic environment, the concentration of solutes outside the cell is lower than the concentration of solutes inside the cell. And this is important because that means we'll have a net flow, a net diffusion of water from outside of the cell into the cell, right? So we're going from higher water concentration to lower water concentration, right? And so as the water continues to move into the cell, you can imagine the cell's like a very delicate water balloon. The water pressure is going to build up inside. And remember, that little cell membrane is so delicate, right? So what's going to happen eventually? The water pressure builds up and causes the cell to break open, to lyse. And what caused it? Osmosis, that movement of water. So this is a danger that all cells face when they live in hypoosmotic environments. So the solution that evolved for many, many cells was that the cells evolved an outer layer, an outer protective layer outside the cell membrane, and this is the cell wall. And one of the most important functions of the cell wall is to protect the cell from osmotic lysis, to prevent the cell from undergoing osmotic lysis when the little cell is living in a hypoosmotic environment. So what happens, water will still move into the cell, water pressure will build up, but now the cell wall, because it's so strong, it will exert an equal and opposite pressure. And again, that's going to prevent the little cell from blowing up. So cell walls are really important. And we'll see, folks, that we find cell walls in plants, fungi, and our big emphasis is going to be the bacterial cell wall. So these are great lecture exam question. So how do bacteria prevent osmotic lysis? They're going to synthesize. Synthesize is just a fancy word for to make. So bacteria will make or synthesize strong cell walls outside their plasma membranes, outside their cell membranes. And it's really important that we know what the bacteria cell walls are made of and what makes them so strong. So the component in bacteria cell walls that makes the cell wall so strong is called peptidoglycan. And remember, folks, only members of domain bacteria can make peptidoglycan. So might ask you just a couple of questions here. So do humans cells have cell walls? And the answer is no. Remember, we're members of kingdom Animalia, and part of being that kingdom is you do not make cell walls, right? So that means that we don't make peptidoglycan. And this is wonderful because that means we can use peptidoglycan as a unique target for magic bullets. Remember Paul Ehrlich and his magic bullets? We're going to find drugs that will specifically bind to or inhibit synthesis of peptidoglycan. That's going to cause the cell wall of the bacteria to weaken, and then the bacteria will die from osmotic lysis. That's what we want if we have bacteria causing harm in our human patients. But to understand how these antibiotics work, we have to understand the structure of peptidoglycan. So let's dive in there. So first of all, the glycan here, this is suggesting that we have some carbohydrate portion. And the carbohydrate or sugar portion of peptidoglycan is made up of two modified glucose molecules. And you might remember from the Unit 2 chemistry, we talked about these two modified glucose molecules. One of them is called NAG for N-acetylglucosamine, and the other one is called NAM for N-acetylamuramic acid. Now, of these two modified glucose, organisms other than bacteria can make NAG. Remember, a polymer of NAG is called chitin, and we find chitin in fungal cell walls. We find it in the exoskeletons of arthropods. So NAG's not unique to bacteria. The molecule that is unique, that only bacteria can make, is N-acetylamuramic acid, right? So because only bacteria can make NAM, only bacteria can make peptidoglycan. So what we'll have in peptidoglycan is a chain of alternating NAG, NAM, NAG, NAM, NAG, 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 NAM. And folks, check this out. This is one of those beta-glycosidic links that we saw in cellulose, remember? We also see them in chitin. And this makes for long, straight chains of these sugar backbones, and that means the chains can pack tightly together. And you might recall when we were talking about cellulose, it was because the chains of cellulose were long and straight, that they could make lots and lots of hydrogen bonds. Well, bacteria take that strengthening to the next highest level. And what bacteria do is to NAM, they synthesize a short little, this is actually quite good. The bacteria attached to NAM, they're going to synthesize a short little amino acid tail. We call this the tetrapeptide tail. And check this out. Here we see the normal L isomers of amino acids, but here we see those really unusual D optical isomers of amino acids. So it's in peptidoglycan that we'll see those unusual D isomers of amino acids. I think I misspoke there. These are the D isomers of the amino acids that are found in peptidoglycan. So in this cartoon, folks, this gives us a better idea of a strand of peptidoglycan. So we go NAG, NAM, NAG, NAM, NAG, NAM. Here's a beta-glycosidic bond, and notice that every other sugar is flipped 180 degrees, just as we saw in cellulose, forming these nice long straight chains. The NAMs will have the tetrapeptide tails here, and you might say, well, what good are those tetrapeptide tails? Well, this is where it's awesome. The bacteria are going to form strong covalent bonds between the tetrapeptide tails on different peptidoglycan strands. So let's move to the next PowerPoint and see if we can see that. So again, we're kind of changed our scale here, you guys. Here are the alternating backbone of NAG and NAM. There. And we can see we have multiple layers. These little balls right here, that's the tetrapeptide tail right here and right there, down there. And this is a cool thing that bacteria do. They form these covalent bridges between the tetrapeptide tails on different peptidoglycan strands. And it's these bridges, these peptide crosslinks, these peptide bridges that make peptidoglycan so strong. So if we could somehow prevent those peptide crosslinks from forming, what would happen to the strength of the peptidoglycan, right? Well, if these peptide crosslinks don't form, that's going to really weaken the peptidoglycan, and as a consequence, if the bacterium is living in a hypoosmotic environment, the peptidoglycan is weakened, the cell wall is weakened, the bacteria is going to undergo death by osmotic lysis. So how could we humans prevent that peptide crosslink from forming? Well, if we knew the enzyme, the bacterial enzyme responsible for forming that crosslink, if we could find a drug that would bind to that bacterial enzyme, we could stop those crosslinks from forming. So let's use the name for the enzyme, you guys, that causes the crosslinks to form. We'll call it bacterial transpeptidase. Okay, so we're going to say the enzyme that catalyzes formations of the peptide crosslinks in peptidoglycan is bacterial transpeptidase. So let's see if we can find some drugs that will stop the function of bacterial transpeptidase. So here's the steps, you guys. This is kind of like physiology. It's like step 1, step 2, step 3. So let's walk through this because this is a lot of information. So first of all, you guys, if I ask you what's the bacterial enzyme that catalyzes formations of the peptide crosslinks in peptidoglycan, you would choose bacterial transpeptidase. When you're doing your readings, you guys, sometimes they're referred to as PBPs for penicillin binding proteins. We're going to use the name transpeptidases. What's the function of the transpeptidases? They form the peptide bonds or crosslinks between the amino acid tails of peptidoglycan. Why are the crosslinks important? The crosslinking is essential for the strength of the cell wall. What would happen if the crosslinks weren't formed? It would weaken the peptidoglycan, and as a result, the cell would undergo osmotic lysis and die. And again, you guys, what's to me is just so cool. A lot of our antibiotics will interfere with transpeptidase. And we'll be moving up to that here soon. Oh my goodness, you guys, sorry again, poor quality, but again, this is a cartoon, a diagram you might see on your lecture exam, or a diagram similar to this one. So again, folks, this is just a detail of peptidoglycan. So here's one strand of peptidoglycan, here's another strand. So questions I could ask you if I put a diagram like this on the lecture exam, I could ask you, what is this? What is this a diagram of? So you're going to tell me peptidoglycan. I could ask you, where will you find it? You're only going to find peptidoglycan in the cell walls of bacteria, right? Okay, and what else can I ask you? Let's take a look here. All right, so another thing I could ask folks is I could ask you to identify, what is this? What's that? What's that? Okay, so and of course I'm going to white out the label. So you guys, this sugar here is the N-acetylglucosamine, and if we go over here, this is the N-acetyl acid. Now if I whited this out, how would you know this is the NAM? Well, remember you guys, the tetrapeptide tail is attached covalently to NAM. So just look for the sugar to which the little tetrapeptide tail is attached, and you know that will be NAM. So that's how you'll figure out what that is. If I circle this and ask you what's this, you're going to tell me it's a tetra. Tetra means 4, tetrapeptide, 4 amino acid tail here. If I ask you what these bonds were, those are peptide bonds, right, between amino acids. If I ask you what bond is this, you're going to tell me this is a glycosidic bond, right, and specifically a beta-glycosidic bond, right here. All right, and what else could I ask you? Oh, now check this out, you guys. Here's a covalent bond between amino acid 3 of one tail and amino acid 4 of another tail, tetrapeptide tail on another strand of peptidoglycan. This is the covalent bond that bacterial transpeptidase forms. This is the peptide crosslink, essential for strength. So if I had a bunch of different bonds labeled, and I asked you which bond will not be formed in the presence of an antibiotic that will inhibit transpeptidase, this is the bond you want to indicate. This is the bond that won't be formed if our bacterial transpeptidase is not functioning. And another question, folks, I could ask you is, we'll be talking about an enzyme that humans make, animals make, called lysozyme. Lysozyme was also discovered by Sir Alexander Fleming. Lysozyme is going to hydrolyze this glycosidic bond. And the result will be the same. The peptidoglycan will weaken, the cell wall will weaken, and then the bacteria will die from osmoticlysis. Um, Animals make lysozyme to protect themselves against invading bacteria. All right, okay. And then down here, folks, we'll come back to this. This is cross sections through 3 different types of bacteria cell walls. And we'll come back to this because we want to be able to identify these cross sections and the components of the 3 different types of bacteria cell walls. Okay, so let's go forward here. All right. So we've been saying that if we could somehow stop the function of bacterial transpeptidase, we could kill disease-causing bacteria from osmotic lysis. So let's find out which antibiotics will inhibit bacterial transpeptidase. So the most famous example would be penicillin. So penicillin is what we call a beta-lactam antibiotic based on chemical structure. And I'll give you the fancy description here. So beta-lactam antibiotics are what we call irreversible competitive inhibitors of bacterial transpeptidase. And an inhibitor is something that stops function. So you want to remember that beta-lactam antibiotics like penicillin, amoxicillin, ampicillin, methicillin, and the cephalosporins, they're going to bind to bacterial transpeptidase and stop the transpeptidase from forming those peptide crosslinks in peptidoglycan. As a result, the peptidoglycan weakens, the cell wall weakens, and you will kill those pathogenic bacteria through osmotic lysis. Another example of an antibiotic that will inhibit those peptide crosslinks from forming. The mechanism is different, you guys. We won't go into the exact mechanism, but just remember that vancomycin, this is kind of a high power antibiotic. Vancomycin will also prevent cross-linking of peptidoglycan and bacterial cell wall. So again, we can kill by osmotic lysis. Just really quickly, you guys, up here, penicillin, the first antibiotic, was discovered by Sir Alexander Fleming. And I know it's a little bit hard to see here, but it was kind of like you could say a lucky mistake. The story is he was studying Staphylococcus aureus, which is a huge cause of wound infections. And just like you guys have done, he streaked his culture of Staph aureus over the surface of an agar plate. So this opaque ribbon here, this is Staph aureus growing on his agar plate. And the story is that he inoculated his plates and then went on vacation for a couple of weeks and left his plates incubating at room temperature. Now, when he was inoculating his plates, by accident, a fungal spore snuck in and landed on the surface of the plate. And while he was gone, the fungal spore germinated and started growing this beautiful colony of a fungus known as penicillium, right? We're going to look at that in lab, or we have looked at that in lab already. So when Fleming came back from vacation, he was checking his plates to make sure they hadn't become contaminated, which is what you'll do. And he came across this plate and he thought, oh, sugar, I got a contaminant here. I'll have to throw the plate away. But he held the plate up to the light and he noticed something incredible, you guys. See this little zone right here around the penicillium colony? See how the Staph aureus aren't growing in that zone? We call these zones of inhibition. And Fleming had the prepared mind to guess that the penicillium fungus was making something, the substance was diffusing into the agar, and whatever that substance was, it was killing or stopping the Staph aureus from growing. And again, he had to prepare his mind to think, what if that substance wasn't toxic? What if we could purify it, and then we could give that substance to our patients suffering with Staph aureus infections or other bacterial infections, and the substance would kill the bacterium and thus save the patient's life? Well, his guess was right, his hypothesis was right. And what was that substance called, the substance that inhibited the growth of the bacteria? That substance was named in honor of the fungus penicillium. That substance was called penicillin. And it was our first antibiotic that was discovered. Antibiotic officially is a substance made by one living organism that kills another living organism. And very interesting that the beta-lactams are produced by fungi and that they will kill bacteria. So some people have hypothesized there's chemical warfare going on out there in nature. Other people challenge that. They other people have discovered that some antibiotics actually are working as chemical messengers between bacteria, excuse me, between microbes. So hopefully we'll be able to explore that a little bit more. All right. So um, one of the problems with penicillin, however, it's really hydrophilic. It's relatively big, a big molecule. And we're going to discover that some bacteria, they have a hydrophobic layer on their outer cell wall, and that can prevent penicillin from penetrating the cell walls of a group of bacteria we call gram-negatives. Um, and and again, I'm hoping in lecture I'll have time to describe that then people took the penicillin and they chemically modified it to make semi-synthetic penicillin or semi-synthetic beta-lactams such as amoxicillin and ampicillin. And the beauty of these two beta-lactams is they can cross the cell walls of that group of bacteria that we call the gram-negative. So we'll come back and talk about that in more detail later. Again, we have lysozyme that cuts the covalent bonds between NAM and egg. Lysozyme is found in our tear, sweat, egg white, our cells of our immune system, our phagocytes make it, right? It's part of our natural defenses against invading bacteria. It was discovered also by Sir Alexander Fleming. It's going to cause the breakdown of Petidoglycan, and we're going to get osmoticlysis of our bacteria. So, folks, I'm going to stop here. This will be the end of part 2. And this really just focused on what cell membranes and bacterial cell walls, the antibiotics that can cause damage to the bacteria through inhibiting proper synthesis of peptidoglycan. And the next part, part 3, will be on gram stains and bacterial cell walls. And this is a big unit. So I'll stop this here and we can take a break and we'll start part